Thank you, John, for the introduction, and thank you and all the other organizers for this great opportunity to hear all these other talks and to speak. So this is a small portion of the human genome, and of the 3 billion bases of the human genome, only about 1% is protein coding, and the vast majority is this non-protein coding regions. And there's a lot of interest in studying and understanding this vast non-protein coding portions of the genome from genome-wide association studies, which have been implicating lots of disease-associated variation in this part of the genome without any sort of clear explanation of why these parts of the genome are associated with disease. And it sort of raises a hypothesis that many of these mutations might be associated with regulatory elements. So what I've been interested in is sort of developing computational approaches which can help us better understand this vast non-coding parts of the genome. And I'm going to talk during this talk about two methods. One is some older work on discovering chromatin states, and then I'm going to talk about some more recent work using massively parallel reporter assays to follow up on these regions and dissect them. So, so to motivate the chromatin states, I mean, if you're trying to understand the DNA sequence directly, it can be very difficult. But if we had markers on top of the DNA, like post-it notes that signified important regions, that could give us a clue on what parts of the genome are potentially important, and then we might see patterns in these markers. Like we might see these two markers co-occurring together frequently, and this could be a signal that there's some important reoccurring pattern that could signify some type of regulatory element. And we could see broad domains of marks, and we could see pairs of marks that occur before different domains. And what these markers are that we actually do have access to are histone modifications, and there's some other type of epigenetic marks. But I'll focus primarily on histone modifications during this talk. So the histone modifications are modifications to the amino acid residues of the histone proteins. So the DNA is wrapped around nucleosomes, which are comprised of an octamer of histones. And they can be subject to chemical modifications, which can vary in a number of different ways of which type of histone modifications you can have. So you can have different types of chemicals, methylation or acetylation. There can be differences in the number of occurrences of the modification, and then which specific amino acid residue and on which histone. And then that's where you see notations like H3K4, Me3 saying histone 3, lysine 4, this is a specific amino acid residue, and then saying methylation in 3 copies. And there's some sort of famous histone modifications where they, at a first approximation, can mark different types of regulatory elements, such as enhancers or promoters or repressive or transcribed regions. But what's sort of driven the field is now the way to systematically map these histone modifications across the genome in a cell type specific manner based on chromatin immunoprecipitation with massively parallel sequencing. So it's a similar or basically the same technique that's also used to map transcription factors. So the idea is you have an antibody which can recognize the histone modification, and then you can extract the DNA where that histone modification is present, and then you sequence that DNA. And then you map it back to the reference genome, and you can create density plots across the 3 billion bases of the genome. In a typical experiment, you might have 20 or million reads. So you get a sense of where various histone modifications are across the genome in a specific cell type. And as I mentioned, you don't just have one type of histone modification. There can be dozens of different histone modifications which you could potentially have data mapped for. And I looked at the question, given that we have multiple maps of histone modifications, could we sort of systematically annotate the genome based on all that information of multiple histone modifications by looking at what is their combinatorial and spatial pattern? So what we were trying to do is find these reoccurring patterns of the modifications and also taking into account sort of domains as well, discover de novo what are those patterns, and then once we've discovered the patterns, we go across the genome and annotate each location in the genome to being an instance of that pattern in the cell type. And then we can start characterizing what are these patterns correspond to biologically once we've annotated each location associated with it. So how we actually do this computationally is based on a multivariate hidden Markov model. So we heard about that hidden Markov models earlier in the first talk of this meeting in terms of genotype imputation. Here we're going to apply them in a very different setting. And then it's a multivariate in the sense that the emissions can be a vector of outputs. So we're not just emitting one character at each position. We can be observing different modifications. So this is a cartoon illustration of our modeling approach. So we're assuming that underneath the sort of genome, there's different classes of biological states. They could correspond to enhancers, starts of genes, transcribed regions, intergenic regions. So there's different types of biological classes of states, but we don't need to sort of know a priori what are all the specific classes. And then what we do observe after pre-processing is a binary presence or absence call of each histone modification at a 200 base pair resolution. And what we do is we binarize the data. If there's more reads mapping to that bin than we would expect based on a Poisson background distribution. And the reason we focus on binarized data is to more explicitly model the combinations of marks that we observe and to have interpretable parameters that can avoid overfitting low-level signal variation. So this is the input. And then what we do is we have a hidden Markov model where we're going to learn the emission parameters. And what we do is we model the emissions with the product of independent Bernoulli random variables. So what we're saying is we have some probability conditioned on the state we're in of observing each modification. And then those probabilities are independent conditioned on the state, but they can be different between different states. And then we also have different probabilities if we're in a given state to either stay in the same state or transition to a different state. And more likely, if you're in some state, at the next position, you're going to stay in that same state. So this could allow us to, for example, here we might not have had enough reads to support seeing an H3K36 ME3 modification. And here we also might not have observed any modification. But by reasoning about the neighborhood information, we could observe that or predict we're more likely to still be in the state 5. So that can be inferred through the transition structure of the hidden Markov model. So once we've learned the model, then we can go across the genome and do a max posterior decoding and infer our most likely hidden state. And this is another view of what we're effectively doing. We have a hidden state at each position along the genome, and then we have an observation of the set of histone modifications. And the hidden state at each position will depend on the hidden state at the previous position. And then what we're doing computationally is we're trying to find parameters that is going to optimize a likelihood where we're taking a product over all the chromosomes, we're summing over all possible state paths through the model, and then we have a product of transition probabilities, and then a product of emission distributions, which are going to, again, be a product of these independent Bernoulli random variables. And this is actually optimized based on Baum-Welch, sort of the forward-backward procedures that you had heard earlier. Those types of ideas involves dynamic programming. And then there's issues about how many states you work with and how to actually initialize the parameters for each model. And in terms of the number of states, in practice what we do is we'll often learn models across a range of states and allow the biological interpretation, desired level of interpretation, select the number of states we work with. In some cases, you can spot sort of clear lower bounds on the number of states if there's certain biological concepts that you would want captured and are only captured if a model has a sufficient number of states. So what you see here now is an actual emission parameters on real data. So this was based on CD4T data mapped by KG Zhao's lab, where we applied this modeling approach where the rows correspond to different states, the columns correspond to different inputs, mostly histone modifications, and then the values correspond to what's the probability we would observe that modification in that state, with higher values corresponding to greater probability we would observe that modification, but it's all probabilistic. So once we have one of these models, and we also have transition parameters that I'm not showing you directly, we can go across the genome, assign each location to one of these states, and then start asking, what do these states correspond to biologically? Are these meaningful? All this was learned de novo. And this is an example of one such state where we compared the locations assigned to this specific state with locations that overlap annotated zinc finger genes or locations of a CAP1 binding. And what we saw was a huge and very specific enrichment for those categories in this one specific state. And if we go into the literature, turns out there's a paper published in 2006, which a large part of this abstract was describing effectively the signature of the state, that the presence of these three histone modifications, trimethylation, H3K9, Me3, H3K36, Me3, and H4K20, Me3, so those exactly those three marks with higher probability. And then they were saying lower probability of these other marks. We more systematically show lower probability of all these other marks are associated with sort of zinc fingers and CAP1 binding. So this is an example where by just taking all this high throughput data, applying the appropriate computational machinery, we're able to discover de novo a very specific biological concept. And then other states, for example, this might not have been expected a priori, but we saw a state which peaked in its enrichment very specifically at the end of gene transcripts, which one might not have expected to see such a state. And then what we did was we more systematically went through each one of these states, overlapped it with a variety of different types of external information that we didn't use in learning the models in terms of gene annotations, evolutionarily conserved elements, other maps of transcription factor binding, repetitive elements, and so forth. And we gave candidate annotations to each state, and we broke them down into sort of a group of various types of promoter states, various types of transcribed regions, different types of intergenic regions, and regulatory elements, and specifically like candidate enhancers. And this is where this type of data can be quite powerful, is that you can have potentially functionally important regulatory elements away from any annotated gene, and this becomes one of the most powerful approaches to identify such locations as epigenetic data. And then different types of repressive and heterochromatic elements and repetitive elements as well. And we tried to do quantitative evaluations of this modeling approach. So one thing we took advantage of is there are certain classes of elements which are well annotated in the genome, such as transcription start sites. So we could ask, how well do we recover annotated transcription start sites compared to if we just use individual marks, or if we trained a supervised classifier to predict them, or if we did like a clustering method which ignored the spatial information. And it turned out that we were almost competitive with the supervised classifier, which had the knowledge of the annotations and wasn't working de novo, and we're outperforming other approaches when we sort of order our states by the sort of optimal ordering. But what's sort of more important here is that because we have this de novo approach, we not only sort of learned the concept of a transcription start site, but we learned different subclasses of transcription start sites. So we found that we had different states that heavily enriched for transcription start sites, but then some were transcription start sites that associated with embryonic development genes, but not these other biological processes, while we had another state which heavily enriched for T-cell activation genes, and this was in T-cells, but not these other categories. So this is something you would sort of miss out on if you were just building supervised classifiers to train on concepts you already know. So what I talked to you about so far was all in one cell type, but one of the sort of powers of the epigenomic data compared to the genomic data is that in different cell types, you're going to have variation in where these epigenetic marks are present compared to the DNA sequence, which is more or less constant across cell types. So if we go into different cell types, we could see the marks change or move around where they're present. And this can give us a lot of information about cell type specific behavior. So the work at the time was based on a compendium of 9 histone modifications and related marks, and then across 9 human cell types that were designed to be very diverse in their coverage. So this is looking at the data at one specific location. The different colors correspond to different cell types, and then different marks within each cell type is shown in the different rows. So this is all at one specific location, and what we wanted to do was have some systematic way to sort of annotate the genome in multiple cell types based on this information. So conceptually, what we did was we applied the same modeling approach, but effectively concatenated the different cell types as if they were additional chromosomes. So we had one common model, chromatin state model across the different cell types, but then we had cell type specific annotations. So we had fewer marks, and we had in this case, we focused on a model with 15 states characterizing different classes of promoters, enhancers, insulator, transcribed, repressive, heterochromatic, or repetitive elements. And again, we overlapped it with external information to help in this characterization. So this is another illustration of what we did here is we have, in this case, the same genomic loci in 4 different cell types. And this is showing you the raw signal, input signal of the different histone modifications. And then we have a chromatin state call in the 4 different cell types I'm showing you here, color-coded based on these annotations. And this is showing you now across 9 cell types. So what somebody can do is they can go into the genome browser and then bring up some location they're interested in, and then quickly scan across some position and see the chromatin state annotation in that cell type, or do computational processing of the dynamics of these chromatin states across the cell types.
Are some cell types more similar? Like, is that in the extra?
Yeah, I mean, it's going to be, so in the modeling, each cell type is sort of treated as independent. It's not trying to learn structure between the cell types, but it is the case that some cell types are more similar to other cell types biologically. And yeah. So I mean, you can see, for example, here, this is a ES cell which has a poised promoter, and then we have these cell types which are all active for this gene, and it can vary depending on what gene you're looking at. So going back to some of the original motivation I had mentioned was we're having these genome-wide association study hits falling in these non-coding portions of the genome, often without additional supporting evidence. So at the time, what we did was we went through the GWAS catalog and a couple other studies, and we overlapped the set of top lead SNPs that were annotated with the phenotype based on the GWAS, and we compared them with our chromatin state annotation. So what you're seeing here, the columns correspond to lead SNPs that were genome-wide significantly associated with the phenotype. The rows correspond to the various cell types, and then I've color-coded the boxes based on the chromatin states as indicated here. And what you can see is that there's this large number of SNPs that are associated particularly with the strong enhancer state relative to what would be expected in other cell types. And if you're familiar with the biology of these 9 cell types, K562 is, of these 9 cell types, the one that would make sense for something that was looking at erythrocyte phenotypes associated with hypertension and cardiovascular disease. And we found a number of other cases just by looking at the lead SNPs where we, when we compare them to what we were annotating as strong enhancer chromatin states based on this data, where, for example, we saw studies that were looking at immune-related diseases in our lymphoblastoid cell type. And we saw things looking at cholesterol and lipids associated with HepG2, which is a liver-related cell type, and then the erythrocytes with the K562 study. So that was all in 9 cell types. I worked as part of the Roadmap Epigenomics Consortium, which sort of took this modeling approach sort of an order of magnitude greater in terms of the depth of the cell and tissue types, and also had a much greater focus on primary cell types opposed to the cell lines. So this was showing you 127 reference epigenomes. 111 were mapped by the Roadmap Consortium, and 16 from the ENCODE project, which were processed computationally through the same pipeline as the Roadmap had. And what I'm showing you in blue are cases where we had the mark of the column and the cell or tissue type of the row with the data present. So we had, in this case, based on these reference epigenomes, we had 5 histone modifications that were mapped where we had at least one data set in all the samples for it. And then this is showing you a screenshot of the chromatin state annotations based on a model learned with 15 states based on these 5 marks. And then we had another model which we added an additional mark, H3K27 acetyl, which can be associated with more active regulatory elements, but that was only mapped in 98 cell or tissue types. So the chromatin states were defined on those 98 for which we had data based on the 6 marks. But as you can imagine, if we kept on adding marks and we wanted to include each cell type which had every mark mapped, we would start quickly diminishing the number of cell types for which we could do this for. So I had come up with an alternative approach where we would first impute the epigenomic data using the available knowledge we had in the compendium based on the correlations with other marks in the same cell type and that mark across the cell types. And if this is sort of a plug to come to my next talk, it'll be Monday, August 1st, this time, where I'm going to talk about some of the details of the computational approach for how we went about imputing the epigenetic data. And then we could learn chromatin states more consistently based on more marks and more cell types. And this is showing you, in this case, we learned a model based on 12 marks with 25 states and an image of this annotation. And now another plug for work. Adriana has been taking the same computational modeling idea, but applying it to completely different data. So if you come to her talk, she'll talk about how we've now been applying chromHMM to annotate the genome based on multi-species alignment data and learning conservation states based on some combinatorial patterns in these alignments. And she'll tell more about that. So that's on Monday, her talk. So now to go to the second part of my talk. So this is about high throughput dissection of regulatory regions. So we now have, based on this epigenomic data, we're becoming effective at generating tens of thousands of predictions in a cell type of candidate regulatory regions. And this sort of calls out for the ability to functionally test in high throughput large numbers of regulatory elements, and also to potentially further dissect these at a very fine resolution of where the important activating and repressive nucleotides are, and to distinguish what's potentially activating and repressive. So what's the enabling work for what I'm going to tell you about is a massively parallel reporter assay. And this has been an ongoing collaboration I had with Tarjei Mikkelsen, which started when we were both at the associated with the Broad Institute. A number of other groups have developed related technology. So the technology that my experimental collaborator Tarjei had developed was this idea that you could synthesize in tens of thousands or now more than 200,000 DNA sequences that you would want to test. You can actually write out explicitly the DNA sequence that you want to test. It's limited to about 145 base pairs, and it's based on a sort of Agilent array, and then they extract the DNA sequences, put them in front of a reporter gene, and have a unique barcode downstream of the reporter gene in like the 3' UTR region. So then if that sequence you designed is able to turn on the reporter gene, then you would sequence that corresponding barcode. So then you can get a count of how much mRNA you have with that specific barcode and how much input you had as well, and then figure out how well that sequence you designed is able to drive gene expression. So in the original paper, which published this technology, what they did was they took two well-known enhancers and they did a systematic mutagenesis. They took each nucleotide, mutated in each possible way. They did multiple different barcodes to control for reduce the noise. But because they were devoting so many sequences to interrogating these two locations, they were only limited to testing two regulatory regions. We had another paper in follow-up paper where we used the same technology and we tested more than 2,000 of the regulatory regions predicted based on the chromatin states, and also did targeted mutations to regulatory motifs. These are patterns that the transcription factor is expected to bind, and confirm that both the large fraction of the regulatory regions were showing activity and that the targeted mutations could disrupt the activity. But what we're interested in now is to effectively combine the elements of these two previous studies to, one, de novo dissect what are the important regulatory bases and discover activating and repressive sites, and to test thousands of locations. And the challenge here, the constraint, is that we have this finite budget on the number of sequences we can test. So we need to be efficient in how we allocate our reporter sequences to both get this high resolution information and to still be able to test large number of regulatory regions. So the design strategy that we converged upon turns out to be a fairly simple design, but if you analyze it the right way computationally, it turns out to be quite powerful. So what we do is we take a regulatory region we're interested in testing, and I'll tell you how we selected those regions soon, and we tile them with 31 reporter sequences in 5 base pair increments. Each reporter sequence that we're testing is 145 base pairs. So we have 295 base pairs of the regulatory region. This has several advantages. One is because we're only devoting 31 sequences per location, we can now test close to 8,000 regulatory regions in a single experiment. We have coverage of 295 base pairs, which is important because from the chromatin data, we can often be confident at a 200 to 300 base pair resolution where the regulatory site is sitting, but it's hard to be confident that this 145 base pairs is the location to test. This is all under the assumption that there is 145 base pairs sufficiently long, which in some cases will be true. If you need longer, then it's a limitation of this assay. We both get robustness to noise because of some of the redundancy of the overlaps, but that's also the differences between some of these reporter sequences is also what can give us access to higher resolution information. And there's also simplicity in this design that we don't have to design a lot of mutations. So the specific data I'm going to tell you about was based on this experimental design, and all the experimental work was done by my collaborator Tarjay's group. So what we had done was we selected about over 15,000 regulatory regions to test. All of them were a DNA hypersensitive site. So this is an assay that measures open chromatin, specifically where the transcription factors are most likely to bind. So it's a higher resolution information. And then we split this based on originally 4 cell types, and we did a stratified random sampling of the different chromatin states. So one of the goals was to understand the differences between different chromatin states in terms of their activity in this assay. And another thing it allowed us to do was to also selectively enrich for testing more cell type specific enhancers, which were potentially some of the more interesting locations to test. And we ended up doing 16 experiments. So we tested all the locations in 2 cell types with 2 different types of promoters, a minimal and a SV40, which has more basal activity potentially. We had 2 sets of sequences based on the same design strategy, but they were different sequences, and then everything was done in replicate. So this is an illustration of the design here of what we're testing at one location. So we have this 295 nucleotide long sequence of letters that we're testing. And then these are 145 base pair subsequences in these 5 base pair offsets. And gray represents basal expression. So each row corresponds to one of the tiles and what positions we're testing. And we're always anchoring at the center of a DNA site. Now, suppose in a very idealized situation that we had an activating site sitting right here that was sufficient to turn on the gene. And then what we could expect is that we might see these tiles have high activity, but all the other ones that were not overlapped it have basal activity. And if it was over here, we expect these to have higher activity and these to have closer to basal activity. If we had an important repressive sequence here, what we might expect are these tiles to have lower activity than even basal. In practice, it's not going to be that deterministic. So what we need to do is have a computational probabilistic modeling approach to take this type of data and to come out with an output like this, which is, and this is actual real data now from the experiments, of our prediction of the relative regulatory activity at each position along the sequence. And what I'm showing you in black is our overall prediction, and then green and blue are based on 2 different promoter types. So it's a little more stringent than replicates because we're changing one variable in terms of the promoters. But you see good agreement there. And then this is showing you another location where we have a prediction of a repressive activity. And if we compare this now with locations predicted to have a regulatory motif site in that cell type based on the centipede method, this was something that came out of Jonathan Pritchard's group, which uses DNA sequence information and also DNA. What they do is they predict there's a regulatory site over here and regulatory sites over here. So this is based on orthogonal information, sort of requiring prior knowledge of the motifs. And what we're able to do is de novo predict that there's regulatory activity here and further make this additional prediction that here it's working as an activator, here it's working as a repressor. And this is something you don't get when you're just focusing on sort of motif calls or DNA footprints. So how do we go about actually doing that inference that I showed you, going from that tile data to this high resolution prediction of where the activating or repressive sites are? So we have this probabilistic model here where what we do is we have a variable for each reporter tile corresponding to its regulatory activity. And then we have another variable for each 5 base pair interval corresponding to the latent hidden activity of that specific position. So we observe the activity of these 145 base pair sequences, but what we would like to infer is the regulatory activity associated with each 5 base pair interval, which we don't get to observe directly, but we're trying to infer based on our observations. So we can create dependencies where each y variable is going to depend on the hidden variables it overlaps. So y1 is going to depend on h1 through h29, y2 is going to depend on h2 through h30, and so forth, where y31 will depend on h31 through h59. And then we make some additional modeling assumptions. We assume that the y's are normally distributed with their mean set to the mean of the incoming h variables and a variance parameter which we set to the empirical variance of the observed data. And then we put a normal prior on the hidden variables where we set its mean to the empirical mean, and then we have one free parameter, which is the prior of the variance of these, which ends up controlling the overall smoothness of the inference. And what we do in practice, and I'll illustrate this next, is we actually try sort of a loose and tight setting of the variance parameter. And at any specific position, we take the more conservative value after a z-score transformation. And then in order to get to single base pair predictions, we do a linear interpolation from this 5 base pair resolution. So just to make the point about this one free parameter which controls the overall smoothness and the trade-off involved, if we set this prior to 1, we would have gotten this value, which was potentially underfitting some of the structure here. If we set it to a higher value, what would happen is sometimes we can see potentially strong activation and repression in the same loci. So what we end up doing is we take the more conservative output at each position, so that avoids this underfitting and also potentially overfitting here. So now this is showing you more examples of our inferences. I'm taking every location that we tested that had a GAB-PA predicted regulatory site active in the cell types by the centipede method in red, blue or other regulatory motifs predicted by it, and black is our overall inferred activity. So you can see it tends to be above average, and sometimes the peak of the regulatory activity is where that motif is sitting. This now contrasts if we take another motif, NRSF, which is a known repressor. The inferred regulatory activity is almost always below average, and quite often you see a strong dip of the inferred regulatory activity right where that motif is sitting. And here's another quantitative evaluation, again using the centipede calls as the orthogonal validation, and showing as we go to greater activating activity or greater repressive activity, we have a greater percentage of the bases overlapping centipede motifs. So we've bin this into bins of 928 nucleotides. And it's greater than both the overall baseline, black is expected. And if we just look at the center position where we both have more statistical power and expect more regulatory sites, that's its baseline, and we're better on, based on either baseline. So that was looking at sort of large aggregation of different motifs. We also looked at individual motifs. Now we're using a set of motifs that were called in a cell type independent manner to compare the difference between these two cell types. So each dot here you see corresponds to one regulatory motif. And on the x-axis is what's its average regulatory activity in HepG2. On the y-axis, what's its average regulatory activity in K562 cell types. And you can see we see two motifs that were consistent activators here. This is showing an aggregation plot, a consistent repressor. What was interesting was one of the strongest activators in both cell types correspond to this 10-mer motif, which has come up in a number of contexts as being one of the most conserved evolutionarily and associated with the highest expression in vivo. But there's still some uncertainty about what protein's actually binding this motif. And if we look off diagonal, we can see examples of cell type specific activators, or even in some cases cell type specific repressors as well. So that was looking at the motifs in average. And another question that sort of comes up is, are there some motifs within a given cell type that are working as both an activator and a repressor? So what we asked was, did we see an enrichment of the motif instances above an activation threshold and below a repression threshold? So the x-axis is showing you the negative log base 10 p-value for being a repressor and the y-axis for being an activator. It turned out most things fell on the axis. We had a large number of activators, some repressors, but there was a few compelling cases of both. We did see some examples like the MAF family, which are known to be involved in both. But for most cases, when we're looking in the specific cell type in this experiment, most motifs had a clear preference for one or the other. And then another thing which we did was we compared it to repetitive elements, and it turned out that the sequences that had some of the strongest activation in this assay were ERV1 repetitive elements. So there was a strong enrichment for those in our highest activity locations, while other repetitive elements such as line didn't show activity in, or enrichment for either repression or activation. And then part of our design was that we could compare the different chromatin states because we had randomly selected DNA sites for each of these different chromatin states. And what we saw was that what we were characterizing as our active promoter or active enhancer were among the most activating states. So these were associated with an acetylation mark, which was associated with activation. And this is outside of the native context. So even though we're testing them on these plasmids, the endogenous chromatin state call still is reflective in the relative activity on average of these different sequences we're testing. And then the last analysis I'll just show you here is one other thing which we can do is we can take individual motifs and compare what's its average activity we observed and what is expected based on the chromatin state that it was observed in. So what you're seeing here is, in this quadrant, active motifs that are found in active context. So this is on the x-axis, the expected regulatory activity based on the chromatin states. The y-axis is observed. But then we found cases where some motifs would sort of greatly outperform what's expected based on its chromatin state context, like p53 sequences were among the most activating, but weren't really strongly associated with the most activating chromatin states, while RFX motifs were found in some of the most activating enhancer or promoter elements, but had sort of well below average activity. So this is a case where you have a repressor or staller motif sitting in some types of enhancers, which would be hard to sort of identify using other types of approaches. So just to summarize, I showed you a chromatin state approach for discovering epigenomic signatures and can be used for cell type specific gene annotations. And then a massively parallel reporter assay, which allows us to dissect these regions and identify at a high resolution where the activating and repressive sites are sitting. And then to acknowledge a lot of the collaborators involved in the work, and a lot of this was done in the context of either the roadmap or ENCODE consortiums. And then this is my lab, and you'll hear more from them if you stay for the longer program. Thank you.